For any engineered plasmid, we need a selectable marker, which helps a genetically modified organism to retain its functionality. Here, I will explain why and two ways of doing it: antibiotic selection and auxotrophic selection. First, let's assume that we have a plasmid without any selectable marker. Here we have the blue and green host genome as well as our red engineered plasmid. This engineered bacteria will be growing, dividing, and competing with other neighboring organisms for resources. When it divides, it may or may not retain the plasmids. Those bacteria that retain the plasmids will express them, which costs a lot of energy and nutrients from the cell. The bacteria that do not express the engineered plasmid don't need as much nutrients and therefore are more efficient at dividing given the available resources. So this puts the organisms with the engineered plasmid under selective pressure and will be outcompeted. One way to prevent this is to add an antibiotic resistance gene to the engineered plasmid and then grow these bacteria in media containing the antibiotic. If grown without any antibiotic, we have the same selective pressure as before, and the engineered plasmid will not be retained. If grown with antibiotic, the engineered bacteria will have a selective advantage over the bacteria without the plasmid, so the plasmid will be retained in the bacteria, which is what we want. This method works, but the bacteria must also be maintained in the antibiotic-containing media, which is not ideal for all applications. Another method is an auxotrophic selection marker. To do this, we knock out a gene, shown in green, that contributes to the production of some essential nutrient. This makes the bacteria reliant on an exogenous supply of this essential nutrient. Then, we place this gene on the engineered plasmid and transform as normal. Since this plasmid supplies the genetic material necessary for producing this essential nutrient, the engineered bacteria is no longer dependent on an exogenous supply. Then we grow the bacteria in media lacking this essential nutrient. If the media does supply this nutrient, then again we see the same selective pressure that was our original problem. However, if we don't provide this nutrient, then the bacteria retaining the plasmid will have the selective advantage because they have this extra plasmid, and the bacteria without the plasmid won't grow because they're lacking this nutrient. To recap, two common options for selective markers are antibiotic selection markers and auxotrophic selection markers.